turn out so well, so we're going to re-inject clone 35 and I believe 13, which show strong positive bands.
Because they give you the best signals.
Correct.
My name is Daniel Tenen. I'm professor of medicine at Beth Israel Deaconess Medical Center and Harvard Medical School, and I'm the director of the blood program of the Harvard Stem Cell Institute. So those of us who work in blood stem cells think that we have a very unique position. And the reason we have this position is that we know more about blood stem cells than any other type of stem cell in the adult human body. The reason that blood has been easier to work with than most other kinds of stem cells is that blood is readily available, both from the blood that's circulating in your arteries and veins, but also it's relatively easy to get material from the bone marrow, which is where the blood cells are produced. The way that we separate stem cells from the blood is we use antibodies that recognize different kinds of surface receptors and antigens on the blood cells, and there are hundreds of them. And by using combinations of those antigens, we can purify blood stem cells down to the point where in our assays 1 in 2 or even fewer score positive for stem cells in those assays. There's no other tissue where we can get that kind of purity. One area of research that we're quite excited about is trying to understand pathways of what we call self-renewal, the ability of a stem cell to renew itself. And that actually is the major focus of the Harvard Stem Cell Blood Program, is try to identify those pathways and then develop ways of augmenting those pathways so we can make more normal stem cells. One area where that might be quite useful would be, for example, the use of cord blood transplantation. So cord blood could be, we don't have enough good matches in general for bone marrow transplantation. Unless you have an identical sibling, it's sometimes hard to find a good match. Cord blood offers the opportunity for almost unlimited matches because you don't need to have quite as good a match in terms of the transplantation antigens for cord blood. The problem with cord blood is there aren't enough stem cells in a typical cord sample. And people have been using 2 cord transplants now, but even there, it's often limiting. And so if we can augment the number of stem cells, that would be a major advance. One example actually has emanated from our own blood program in terms of trying to augment stem cell function by understanding self-renewal. That came from a screen performed by Len Zahn in our group in zebrafish, where I identified a specific pathway involving prostaglandins that from the zebrafish screen, he went to mouse studies to demonstrate that it enhanced murine stem cells, and now there's a clinical trial with cord blood transplantation being used with that particular compound. There's a lot of interest, a huge interest right now in the cancer field, in the cancer stem cell. Most of the studies in that have been done in blood systems and involve leukemia models, for the reason that we know how to purify normal hematopoietic stem cells very well, and we can purify these cancer stem cells or leukemia stem cells very well. So although officially we're working in normal processes, it's very clear that what we're studying will be highly relevant to leukemia and other kinds of cancers. Another major effort is to organize data that has already been generated in a number of the Harvard Stem Cell Institute labs and is currently being generated around the world in terms of looking at genes involved in hematopoietic stem cells in a way that investigators can ask questions about specific genes across many different data sets, and secondly, can compare different data sets to identify genes in common, to identify the most important regulators of stem cell function. The bioinformatics support to do this was totally enabled by HSCI. It was something that was funded as part of the blood program. And I think it's really taken a life of its own because people are so enthusiastic about it. The Harvard area is incredibly rich in outstanding investigators in hematopoiesis and leukemia, which is the obvious corollary. one thing that HSCI has done is further unite these different investigators in terms of working together. And I think there's actually a very good, genuine spirit among the HSCI. And so participating in that, I think, brings us closer together.